Hello everyone at CEoT. I am Eric Weimer. I'm an associate professor at UNC Chapel Hill, and I am also the associate director of our HLA laboratory.
And I'm Katherine Newhall, an associate professor of mathematics at the University of North Carolina at Chapel Hill. By combining our expertise, we developed an AI-driven approach to virtual cross-matching. This is a new application of machine learning software that enables the assessment of the donor-recipient pairs and allows it to be done more quickly, more consistently, and at unprecedented scale. As you know, the histone compatibility of the donor-recipient pair is correlated to the transplant outcome. At its core, the program uses donor and recipient data, and it quantifies this risk of a positive cross-match and thereby organ rejection.
Our project has accomplished a number of goals. Some of those are increasing the speed and scale at which histocompatibility and eplet matching can be performed. We have increased the accuracy of virtual cross-matching prediction in a number of ways. We can do this by refining the ability to list unacceptable antigens on an individualized or personalized way. Both of these combined together enhance patients' access to organs and give us greater insight into the transplant immunobiology that's occurring.
The project began with us combining our divergent backgrounds to apply mathematical modeling to immunobiology problems. Our initial results demonstrated that these statistical models could predict what the humans were already doing and the physical cross-matches. Then, in 2020, we partnered with Microsoft to harness the power of machine learning algorithms.
Through our project, we developed 3 separate machine learning algorithms trained on our historical data. The first is sort of the model that all of you are familiar with. It utilizes patient HLA antibody data combined with donor HLA typing data. This approach achieves approximately 91% accuracy in predicting a physical cross-match outcome. The second and third models that we developed actually make use of the patient and donor HLA typing data, convert that to HLA eplets, and use that combined eplet mismatch to determine a physical cross-match outcome. What's important to highlight is the second and third models do not use patient HLA antibody data at all. That individual model can achieve a 98% accuracy in predicting a physical cross-match. So by combining these approaches, we have been able to not only enhance the prediction capacity of a physical cross-match outcome, but now we have a better way of gaining insight into what's occurring between the recipient and the donor.
Today, we are working with partners to put this software and this tool into the hands of transplant centers. We are still in the early development, but we hope to be piloting with healthcare systems as soon as possible. At the same time, we'll be working to refine and expand our research with these models. AI cross-matching is more than just a tool. It's a platform. It's a starting point for research and new clinical approaches. For a start, it will allow virtual cross-matching to be expanded to new organs like the heart and lung, where in the past, there wasn't time to do a cross-match. Now, we can scan through thousands of possible recipients to find the right one quickly.
But we can also invert this process and say we have a patient, we have their HLA antibody data, we have their HLA typing data. What is the immunology of the donor that would give them the lowest physical cross-match outcome?
We are very excited about this potential impact to research as well as the clinical approaches. 5 years in, it still feels like we're in the early days. The project continues and its potential only grows as more transplant centers become involved and work with this tool. We look forward to discussing all of that work with you in the future. Thank you.